Imagine a patient who is given, maybe a year to live with an incredibly aggressive brain cancer.
Right, which is already a devastating diagnosis.
Totally. But instead of succumbing to the disease, they end up surviving for over a decade.
Wow.
Yeah, over 10 years on a completely standard chemotherapy drug. Complete remission. And for years, we couldn't easily explain why.
Because if you looked at their DNA sequence, it looked identical to the patients who didn't survive, right?
Exactly. The actual letters of their genetic code, they held no obvious clues at all.
It's really one of the most profound mysteries in oncology. You have the exact same tumor type, the exact same chemo, and yet totally opposite outcomes.
Right. And that mystery is exactly what we're unpacking today in this deep dive. We've got a fascinating review paper by Avalos, Ferrer, and Esteller.
It's a fantastic paper.
It really is. And the mission today is to completely unpack it because it reveals that the answer to why certain tumors shrink while others just keep growing isn't always found in the DNA sequence itself.
Right. It's actually found in this hidden microscopic layer of chemical control sitting right on top of the DNA.
Exactly. It's called DNA methylation. And according to this paper, it doesn't just diagnose cancer, it actively predicts how a tumor is going to respond to our best therapies.
Which is huge. That invisible layer of control completely changes how we think about personalized medicine.
It really does. But before we get into those incredible clinical cases like the 10-year survivor, we need to set the baseline. What exactly is DNA methylation?
Well, at its absolute core, DNA methylation is just a tiny chemical modification. The cell takes a methyl group, which is just one carbon atom attached to 3 hydrogen atoms, and physically attaches it to the DNA molecule.
But it doesn't just stick these methyl groups randomly anywhere, right? There's a pattern.
Oh, yeah. There is a very specific pattern.
Right.
In humans, this almost exclusively happens to the cytosine nucleotide.
Okay.
And not just any cytosine. It targets a cytosine that is sitting immediately next to a guanine base. We call these pairs CpG dinucleotides. And these CpG pairs aren't scattered evenly. They clump together in dense little neighborhoods called CpG islands.
And those islands are usually located at the promoter regions of our genes, right? Like the starting line where a gene actually gets turned on.
Exactly.
Okay, let me try to visualize this for everyone listening. Imagine your entire DNA genome is the keyboard of a massive grand piano.
I like this.
Right. So the individual keys are your genes. DNA methylation, then, is the sheet music. It tells the cell which keys to play loudly and which keys to just completely ignore.
I'd actually take that analogy even further.
Oh.
It's not just sheet music. It's physical. It's like placing heavy lead paperweights directly onto certain piano keys.
Oh, wow. Okay.
Right. So if the paperweight is there, the cell's machinery physically cannot press the key down. The gene is totally silenced.
I love that. So in a normal, healthy cell, you rarely see these heavy paperweights sitting on the promoter regions. The piano is fully playable.
Exactly. But in cancer, the cell goes completely rogue with the paperweights.
Throwing them everywhere.
It's pure epigenetic chaos. In cancer, you see 2 massive shifts happening simultaneously. First, the cell starts dropping these heavy paperweights, which we call hypermethylation, directly onto the CpG islands of tumor suppressor genes.
Wait, so it forcefully silences the exact genes designed to stop tumors from growing?
Yes. It turns off the brakes. And at the exact same time, you see global hypomethylation.
Hypo, meaning less.
Right. The rest of the genome loses its normal healthy methyl marks, which leads to massive structural instability. The genome literally starts falling apart.
That is wild. Now, if you're listening, you might be wondering why oncologists care so much about these microscopic paperweights.
It all comes down to stability.
Right, because RNA, which also tells us what genes are active, it's notoriously fragile. It falls apart almost instantly in the lab.
Oh, it's a nightmare to work with. But DNA methylation is incredibly tough. It survives the really harsh chemical treatments hospitals use to preserve tumor biopsies, those FFPE samples.
And even better, when a tumor cell dies, it sheds fragments of its methylated DNA right into the bloodstream.
Which is the crucial clinical hook here. Because those paperweights stay firmly attached to the DNA fragments floating in the blood, we can read a tumor's exact vulnerabilities through a simple liquid biopsy.
Meaning we don't necessarily have to drill into a patient's brain or lung to know what the tumor's doing.
Exactly.
Okay, which brings us perfectly back to that patient I mentioned at the start, the 10-year survivor with glioblastoma, or GBM.
Let's unpack that.
Yeah, how does a methyl group tell us if chemo will work? So the standard treatment for GBM is a chemotherapy called temozolomide. What is that drug actually doing?
Well, chemotherapies is an alkylating agent. Its entire purpose is to sneak into the tumor cell and attach these bulky alkyl groups onto the DNA, specifically at the guanine bases.
So it's actively damaging the DNA.
Right, it creates physical roadblocks. When the tumor cell tries to copy its DNA to divide and grow, it hits those roadblocks, the DNA shatters, and the cell dies.
But tumors are, evolutionary survivalists.
Yeah.
They have a defense mechanism to stop this, don't they?
They absolutely do. Tumors utilize a repair enzyme called MGMT.
MGMT. Okay.
Think of MGMT as a highly efficient cellular repair crew. It constantly scans the DNA, finds those deadly roadblocks left by the chemotherapy, and just meticulously removes them.
Wait, so it literally fixes the chemo's damage, making the drug totally useless.
Exactly. The oncologist is pumping in this toxic chemotherapy, and the tumor is just quietly sweeping it away.
That is so frustrating. But here's the vulnerability. If the tumor decides to drop a methyl paperweight onto the promoter region of the MGMT gene,
that repair crew is fired. The gene is silenced.
And that is exactly what happened with our exceptional responder, right?
Precisely. When the oncologist gave the temozolomide, the tumor was defenseless, the DNA damage accumulated, and the tumor was just destroyed.
But wait, I remember from the text there was an extra layer to that specific patient's story. It wasn't just MGMT.
No, it wasn't. And that's the beauty of looking at the whole system. That patient had a rare double whammy.
A double whammy.
Yeah. That epigenetic silencing of MGMT knocked out one specific repair mechanism, the direct repair pathway. But this patient also had a completely separate genetic mutation in a gene called APX1.
Okay. And what does APX1 do?
It controls a secondary repair system called the base excision repair pathway. So with that mutated, both primary and backup repair systems were offline.
Wow. So it was a perfect storm. The tumor lost its primary defense epigenetically and its backup defense genetically.
It had absolutely no way to survive.
That is incredible. And for everyone listening, a really great way to lock this concept in is the acronym MGMT. Just remember, methyl group means treatment works.
I love that mnemonic. And it's actually become clinical dogma now.
Really?
Oh, yeah. NCCN guidelines practically mandate testing for MGMT methylation in high-grade gliomas using assays like pyrosequencing. We have to know if that off switch is flipped.
Okay, let's do a quick recap here. You're about 5 minutes in. First, DNA methylation is adding a methyl group to CpG islands, which acts like a heavy paperweight to silence genes.
Right.
Second, it's highly stable in hospital samples and liquid biopsies. And third, in glioblastoma, a methylated MGMT gene means the tumor cannot repair chemo damage, making it highly treatable.
Spot on.
So here's a question for you listening. Think about your own field of work. What's an invisible off switch you absolutely have to check before starting a major project? Because for oncologists, that switch is MGMT.
It really is the ultimate biological off switch.
So if I'm capturing the essence here, methylation acts as this stable record that tells us precisely where the tumor's defenses are down. But chemotherapy isn't always about smashing DNA.
No, not at all.
What if the drug just can't stay inside the cell long enough to work? Let's talk about the cellular bouncers.
Ah, yes. This is a massive issue in breast cancer when we use drugs like doxorubicin.
Okay, walk us through it.
So the drug needs to physically accumulate inside the cell to kill it. But tumors frequently hijack a gene called ABCB1.
And what does ABCB1 do?
It produces a mechanical pump P-glycoprotein that sits right on the cell membrane. It literally grabs the chemotherapy molecules and throws them back out of the cell.
Like a cellular bouncer. But hang on, I want to challenge the logic of the tumor here.
Okay, go for it.
If a tumor can silence a gene with methylation, and silencing this bouncer gene makes it highly vulnerable to our drugs, why on earth would the tumor silence it? Isn't it shooting itself in the foot?
That is a fantastic question, and it really comes down to evolutionary trade-offs.
How so?
You have to remember, the tumor had no idea the chemotherapy was coming. Running thousands of these microscopic pumps takes a massive amount of cellular energy.
Ah, okay.
So early in the tumor's development, before any drugs were even present, silencing that pump was an energy-saving measure. It helped the cell grow faster.
Oh, so it made an evolutionary gamble to save energy, and that gamble became a fatal flaw the exact moment we introduced doxorubicin.
Precisely. Because with the bouncer silenced, the drug rushes in and stays in.
That's fascinating.
And tumors try to handle drugs in other ways too. Like instead of pumping them out, some try to neutralize them using a gene called GSTP1.
Wait, Neutralize them how?
Well, GSTP1 creates an enzyme that essentially blends the toxic drug with a molecule called glutathione, creating a totally harmless compound.
It's like the tumor's making a detox smoothie to clear out the chemo.
That's exactly how it functions. But if we find heavy methylation on the GSTP1 gene, we know the tumor has lost its ability to make that detox smoothie.
So the drug remains highly toxic inside the cell, drastically improving survival.
You got it.
Okay, we've talked about blunt force chemotherapies, but modern oncology is really moving toward highly specific, targeted therapies, right?
Absolutely.
And the paper discusses this brilliant concept called synthetic lethality. This is where it gets really interesting, specifically regarding the BRCA1 gene and PRP inhibitors in ovarian cancer.
Synthetic lethality is probably one of the most elegant strategies we have right now.
So explain how BRCA1 fits into this.
Well, BRCA1 is a gene responsible for a flawless type of DNA repair called homologous recombination. When a cell's DNA physically snaps in half, BRCA1 helps stitch it back together perfectly.
Okay, and we've known for decades that genetic mutations in BRCA1 cause cancer.
Right, but those mutations also create a massive Achilles heel. If a tumor lacks BRCA1 and we give it a targeted drug called a PRP inhibitor,
Which blocks a backup repair pathway, right?
exactly, the tumor then suffers catastrophic DNA rearrangements and just dies.
So the drug exploits the broken gene. But this review highlights something really wild. You don't even need a genetic mutation to exploit this.
That is the big breakthrough. If the BRCA1 promoter accumulates enough of those methyl paperweights, if it's silenced at like 70% levels or more, the cell entirely loses its ability to perform homologous recombination.
Wait, so the epigenetic silencing perfectly phenacobes the genetic mutation?
Yes, the tumor behaves exactly as if the gene is broken, making it exquisitely sensitive to PRP inhibitors.
The sheet music tells the tumor to forget how to repair itself, even though the physical keys on the piano are perfectly fine. That is mind-blowing.
It really shifts the paradigm.
And methylation doesn't just break repair systems, right? It completely rewires how a tumor eats and communicates.
Oh, absolutely. Let's look at communication first. In melanoma, there is this regulatory gene called TBC1D16.
Okay.
In normal cells, it has healthy methylation. But melanoma cells often strip those methyl paperweights away. The gene turns on aggressively.
And what happens when it turns on?
It fundamentally alters how the cell's growth receptors, like EGFR, get transported and recycled inside the cell's endosome.
It's like the tumor builds a brand new high-speed superhighway for growth signals.
Exactly. It causes a worse prognosis because it makes the melanoma incredibly aggressive.
Building that superhighway creates another trap, doesn't it?
Yes, the tumor becomes completely addicted to that new signaling pathway. Because of that epigenetic rewiring, the melanoma becomes paradoxically highly sensitive to specific targeted drugs called BRAF and MEK inhibitors.
So the tumor's greatest strength becomes its fatal weakness.
Exactly.
And we see the exact same kind of addiction with how the tumor eats. There's a gene mentioned in the paper called SVIP.
Oh, the metabolism rewiring.
Yeah. When SVIP gets silenced by methylation, the tumor panics and drastically alters its metabolism. It shifts into overdrive, relying almost entirely on glucose for survival.
Right, it forces the cell into glycolysis. It becomes a total sugar addict.
And once we can read that epigenetic addiction, we can weaponize it. Tumors with silenced SVIP are remarkably vulnerable to experimental drugs called GLUT1 inhibitors, like BAY876.
Because those drugs block glucose from entering the cell.
Right, because the tumor is epigenetically hardwired to demand sugar. Blocking it effectively starves the tumor to death.
It's a brilliant trap.
Okay, let's bullet this out for our second recap. We're at about minute 10. 1. Methylating pump genes, like ABCB1, traps drugs inside the tumor.
Correct.
2. Methylating BRCA1 creates synthetic lethality, making tumors totally vulnerable to PRP inhibitors without needing a genetic mutation. And 3. Methylation can rewire a tumor's metabolism, opening doors for highly specific targeted drugs.
Perfect summary.
So, question for you listening. If you could look at a tumor's metabolic addictions right now, would you try to fix the addiction or use it as a trap like we do with SVIP?
Using it as a trap is definitely the oncology approach right now.
Yeah, it's so clever. But we've been looking entirely inside the single cell so far. What happens when we zoom out?
To the immune system.
Exactly. How does this internal sheet music affect how our body's massive immune system actually sees the tumor?
This is where we cross into the realm of immunotherapy, which is huge right now. And the interaction hinges on a gene called MLH1.
Okay, what does MLH1 do?
It's critical for fixing typos when DNA is copied. But in colorectal cancer, the tumor often drops heavy methylation onto MLH1, completely silencing it.
Which leads to mismatch repair deficiency, right?
Yes. Basically, the cellular spell checker is turned off.
And without a spell checker, the DNA code just starts mutating wildly.
Right, a state called high microsatellite instability, or MSIH. Because the DNA code is such a mess, the tumor starts manufacturing thousands of weird, abnormal proteins called neoantigens.
So the tumor physically starts looking like totally alien to the rest of the body.
It becomes a glaring beacon. The host's immune system detects these alien proteins and just swarms the tumor.
Wow.
Because of this, finding MLH1 methylation is an incredibly strong predictor that a patient will have a brilliant response to immune checkpoint inhibitors, like pembrolizumab.
And isn't it true the FDA has fully approved immunotherapies based entirely on this specific biological mechanism?
They have. It's a massive success story.
But if I'm putting myself in the shoes of the tumor's evolutionary drive, surely it tries to use methylation to hide from the immune system too.
It absolutely does. The flip side of the coin is global hypomethylation.
Right, losing the paperweight.
Exactly. We all have ancient viral DNA permanently embedded in our genome elements called line 1 that make up a huge portion of our DNA. Normally, they are heavily silenced.
But some melanomas just strip that methylation away, creating widespread chaos.
Yes. But here's the paradox. You'd think chaos would make more neoantigens and attract the immune system even more, right?
Yeah, that's exactly what I'd assume.
But this specific flavor of genomic instability actually helps the tumor evade immune recognition. It fundamentally changes the tumor's microenvironment, suppressing the immune cells.
So you can't just count the number of mutations. You really have to understand the specific epigenetic flavor of the tumor.
Which is exactly why researchers are moving beyond just looking at one gene, like MLH1, and using artificial intelligence to look at hundreds of markers at once.
Oh, right, the multi-gene signatures.
Yeah. In non-small cell lung cancer, we have the epimmune signature and FOXP1 methylation to predict anti-PD-1 responses.
And it goes even further than that, doesn't it? We see this in living drugs like CAR T-cell therapy, where we engineer a patient's own immune cells.
Right. The paper mentioned the EpiCart signature. They found that an epigenetic signature from the patient's T cells before they are even infused back into the body predicts that the therapy will work.
But there was a massive catch with the manufacturing, right?
There was. The longer you culture those CAR T cells in a lab, the more dysfunctional their DNA methylation becomes.
So the manufacturing time in the lab literally ages the cells epigenetically, making them less effective. That's a huge logistical insight.
It changes how we think about producing these therapies.
Okay, here is a wild question. If these methyl paperweights are just chemical tags and they're causing all these problems, can we just use drugs to wash them off?
We can, and we do. We have epigenetic drugs called DNMT inhibitors, like azacitidine.
How do they work?
When the tumor cell divides during the S phase, these drugs sneak into the DNA and physically degrade the methyltransferase enzymes. It effectively erases the tumor's hypermethylation.
Which lifts the paperweights and wakes up the tumor suppressor genes so they can finally stop the cancer.
Yes, but it also triggers an amazing side effect called viral mimicry.
Oh, I remember this.
Right. Remember those ancient line 1 retroviruses embedded in our DNA? By wiping away the methylation, we wake them up?
It's literally like opening Jurassic Park inside the cell.
That's a great way to put it. The tumor cell detects this sudden surge of viral RNA, thinks it has been infected by a massive pathogen, and hits the panic button. This triggers a massive immune response against the tumor.
We literally trick the tumor into thinking it has a deadly virus so our own immune system will attack it. That is brilliant.
It's amazing biology. But tumors are relentless shapeshifters. They fight back.
Yeah, the paper spends a lot of time on lineage plasticity. If you're listening, you might be wondering if tumors can just alter their methylation to survive. How extreme can those changes get?
The changes can be total. I mean, under the intense pressure of our best drugs, a tumor can actively rewrite its entire epigenetic landscape to completely change its cellular identity.
Like change what kind of cell it is.
Exactly. In prostate cancer, under the pressure of androgen-targeted therapy, the tumor can alter its methylation to transdifferentiate into a neuroendocrine tumor.
Wait, it just stops relying on hormones entirely and becomes a different tissue.
Yes. The review also notes cases where B-cell leukemia physically transforms into macrophage-like cells to escape therapy.
It sheds its original identity like a disguise.
It literally physically changes its identity to survive.
Okay, time for our third recap. Minute 15 check-in. 1. MLH1 methylation causes massive mutation burdens, making the tumor a massive target for immunotherapies.
Right.
2. CAR T-cell manufacturing time directly impacts therapeutic success due to epigenetic aging.
Oh.
And 3. Tumors can use methylation to shapeshift and avoid drugs, which is lineage plasticity.
Exactly right.
Which raises a massive question for you, listener, to consider. If a tumor can literally change its identity from a B cell to a macrophage to survive a drug, what does that mean for how we even define a cancer type in the future?
It completely disrupts our traditional diagnostic categories. Are we treating the organ, or are we treating the active sheet music?
Exactly. Which brings us to our final hurdle. If this epigenetic profiling is the key to unlocking personalized medicine, trapping drugs, and triggering viral mimicry, why isn't every single patient getting a full epigenetic workup right now?
Because translating this from the laboratory to the bedside faces several massive roadblocks.
Okay, lay them out for us.
The first is intratumoral heterogeneity. A tumor isn't a monolith. It's an ecosystem of millions of subclonal populations.
Right.
Different pockets of the single tumor have entirely different methylation profiles. If you take a standard bulk tissue biopsy, you get an average signal. The signal is diluted.
It's like listening to a massive choir of 10,000 people and trying to pick out the one single person singing off-key who's going to cause a relapse.
That's a perfect analogy.
Right.
And relying on liquid biopsies looking for circulating tumor DNA or ctDNA has its own problems.
Because it's too hard to find.
Yeah. In early-stage cancers, the amount of tumor DNA in the blood is vanishingly small. Plus, normal hematopoietic blood cells naturally release a ton of biological background noise. Finding the tumor signal is like trying to hear a whisper in a hurricane.
And the actual chemical process we use to read the methylation doesn't exactly help the situation, does it?
No, it doesn't. That's the third roadblock. The gold standard method is called sodium bisulfite conversion.
What does that do?
It chemically alters unmethylated cytosines so we can distinguish them. But the process is brutal. It chemically shreds and destroys the vast majority of the DNA sample.
Oh, wow. So when you only have tiny, fragile traces of tumor DNA from a liquid biopsy.
Right. Destroying 90% of your sample before you even analyze it is a complete disaster.
Okay, but what about the AI? You mentioned earlier we use machine learning to read these massive multi-gene signatures. Why is that a roadblock?
Because of the black box problem. The AI model looks at thousands of methylation spots and spits out a prediction like, this patient has a 90% chance of responding.
But it can't tell the clinician why.
Yeah. Exactly. It lacks explainability. It's recognizing a vast, complex visual pattern of paperweights on the keyboard, but it doesn't know the song. It doesn't understand the underlying biology.
And regulators and doctors are obviously hesitant to make life or death decisions based on an algorithm they can't cross-examine.
Exactly.
So, how do we push past this? Where is the field heading? Give us your final perspective.
Well, we are seeing rapid technological leaps. To solve the harsh chemistry problem, we are moving toward third-generation sequencing, like Oxford nanopore technology.
How is that different?
These devices pull the native, untouched DNA strand through a microscopic pore and use electrical currents to read the methyl groups directly without destroying the sample.
Oh, that's amazing.
And we are also combining multi-omics with single-cell profiling to finally map out that complex tumor choir cell by cell.
So DNA methylation is really transitioning from a static diagnostic tool into a dynamic, real-time monitor for adaptive cancer management.
Exactly. It's the literal bridge between our hard-coded genome and the ever-changing environment.
Which brings me to a final provocative thought for you, listening to mull over. We know that DNA methylation isn't just something that happens in cancer.
No, it happens in all our cells.
Right. It is highly dynamic and constantly influenced by our age, our lifestyle, and our environment. If we know that cancer therapies can actively remodel a tumor's epigenome, and our own daily behaviors influence our overall methylation.
Oh, I see where you're going with this.
Is it possible that our daily environmental exposures are actively writing the sheet music for how our bodies will respond to future medicines? Could we one day actively prehabilitate a patient's epigenome before we even give them the chemotherapy?
That is a completely wild but totally plausible new frontier of preventative oncology.
It really is. Keep questioning the sheet music of your own cells, and thank you for taking this deep dive into the source material with us. We'll see you next time.